Okay, everybody, good morning. Today we'll talk about the muscle tissue. In this lecture, we'll mainly talk about the structure of the skeletal muscle, mechanism of contraction of the skeletal muscle, and a neuromuscular junction, how the nerve talk to the muscle. So, first, you know that there are three types of muscles or muscle tissues are present in the human body: skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. Only skeletal is voluntary. That means you can control the skeletal muscle contraction. And you all know that you move your body parts, right? By using skeletal muscles, and it is voluntary. Smooth muscle and cardiac muscle, those are involuntary. You cannot control them. For example, your heart, right, is cardiac muscle. You cannot stop. If you tell your heart to stop, it won't listen to you, right? Your blood vessels, your GI tract, intestine, esophagus, you cannot close them or open them. They will do their work involuntarily. Here you see how those three types of muscle tissues look under the microscope. You have already seen this under the microscope. So first one you see is the skeletal muscle under the microscope. Skeletal muscle fibers are cylindrical, long cylindrical fibers like this. Multiple peripherally located nuclei. Inside each fiber you have many or multiple nuclei, and they are not located centrally. They are attached to the membrane. Peripherally located multiple nuclei. And also, if you look carefully, you will see striations, lines like this inside the muscle fiber. So that is the skeletal. Middle one is a cardiac muscle tissue under the microscope. Fibers are also cylindrical, but they have branches and usually single nucleus. Striations present. Another thing which is different than other muscles is intercalated discs, thick bands. You see here, these are intercalated discs. So that is one important feature of cardiac muscle. Smooth muscle. The fibers are spindle-shaped fibers like this, like your eye. Both ends are pointed and middle part is wider. Okay, so spindle-shaped fibers, single nucleus located centrally. So very nice. No striation, no branching, no intercalated disc, right? So very simple. So those are three types of muscle tissues, how they look like under the microscope. Now, the characteristics of muscle. Muscle tissue shows some properties. What are those important properties of muscle tissues? Excitability, contractility, extensibility, and elasticity. I'll just explain those. Excitability. If you give any stimulus to the muscle tissue responds to that stimulus, and that is called the excitability. The ability to respond to any stimulus. For example, if I poke your muscle, it will contract, right? If I hit your muscle, it contracts. So that's the ability to respond. Stimulus is you are striking or poking the muscle, that is the stimulus, and respond is the contraction. So how the muscle responds? By showing contraction, and that ability to get shorter is called contractility. The ability to get shorter. And extensibility is the ability to get stretched. If you pull the muscle from both ends, it will expand. That is the extensibility, the ability to expand. It can contract as well as it can expand. If you put pressure, pull from both ends, it will expand. Elasticity. After you see, look at me, after you expand the muscle, if you leave the pressure, then it will get shorter again. Will go back to its actual shape. That is the elasticity, the ability to recoil. After you stretch it, if you remove that pressure, it will get back to its actual shape. So that is the elasticity. So I'm repeating again, excitability is the ability to respond to a stimulus. Contractility is the ability to get shorter or shortening, and then extensibility is the ability to get expanded by pressure, and elasticity is the ability to recoil, to go back to its actual shape. Functions of muscle. Movement of bones. You all know that skeletal muscles are attached to the bones, and by the contraction of the skeletal muscles, we move our bones in different directions. That is the movement. Circulation of fluid. The best example is your heart muscle contracts and relaxes, and that circulates the blood throughout the body. You know that. Maintaining posture and body position. By contracting and relaxing different muscle groups, we maintain our posture or body position. Stabilizing joints. You know that muscles cross the joints, otherwise they won't be able to move the body parts. Muscles cross the joints. For example, look at me. The muscles, those that start in your arm, cross the elbow, right? And get attached to the forearm. The muscles originate in forearm, they cross the wrist joint and go to the hand. Make sense? So, muscles must cross the joint to move the body part. And by crossing the joints from different sides, the muscles stabilize the joints. Generation of heat or production of heat is another important function I have mentioned before. If your body gets cold, what will happen? Shivering will occur, right? And shivering is muscle contraction, frequent muscle contraction that will increase metabolism, and metabolism produces heat in the body. So that's how the shivering increases the body temperature and produces heat. So those are important functions of the muscle. Now we'll see the structure of a skeletal muscle. So if you see a skeletal muscle, and cut the skeletal muscle, this is the whole muscle, okay? This is the whole skeletal muscle. Now if you cut it, you'll see inside the muscle there are columnar structures like columns. These are called fascicles. So these are the fascicles. And inside the fascicles, you have again columnar structures. So these columnar structures are muscle fibers. Muscle fibers. Okay. So inside the muscle you have fascicles. Inside the fascicles you have muscle fibers. And inside the muscle fibers you have thin rod-like structures like this. Rod-like structures. These are called myofibrils. So many tiny, like if you make cross section, you will see like many dots. Those are actually thin rod-like fibers like this, many bundled together. So if you cut, you will see like many dots. Okay. So myofibrils. So this is how the structure, skeletal muscle structure is. Whole muscle, then fascicles, then inside the fascicles you have muscle fibers. Inside the muscle fiber you have myofibrils. Now, if you see the whole muscle, it has a connective tissue covering. Also, the fascicles, each fascicle has connective tissue covering like this. Then muscle fiber also has connective tissue covering. So you see three coverings. One covering connective tissue membrane that covers the whole muscle, another covers the whole fascicle, and another covers the muscle fibers. Okay. So the membrane, connective tissue membrane that covers the whole muscle, that is called epimysium. This is the epimysium. It covers the whole muscle. Then the covering of the fascicle is called perimysium. So perimysium is the covering of the fascicle. Okay, and then the covering of the muscle fiber is called endomysium. Endo means inside, that's the innermost one. Endomysium. So you see three coverings: epimysium, whole muscle; perimysium, muscle fascicle; and endomysium, the muscle fibers. Okay, so that is the structure of a skeletal muscle. Now, here you see they have shown nicely the picture you see here. Now you see inside the muscle fiber you have myofibrils, right? Those are rod-like structures, myofibrils. Okay, so we will see the structure of a myofibril. Okay, that is very microscopic. If you just take one of these thin, rod-like structure, myofibril, out like this from there and see under the microscope how it looks like. So we are just looking one myofibril. So if you see a myofibril under the microscope, you will see many segments in it. Each segment, so this whole thing is the myofibril, Okay, and inside the myofibril you will see segments. Each segment is called a sarcomere. Sarcomere. So this is one sarcomere, this is another, this is another, this is another. So same thing repeated in the myofibril. And the sarcomere is bounded by Z discs. Okay, so this is one Z disc, this is another, this is another. In between two Z discs, the area is a sarcomere. So sarcomere are bounded by Z discs. Now, sarcomere is called the functional unit of muscle. Again, I am repeating, sarcomere is called the functional unit of muscle. That means sarcomere is the unit of contraction, because we know functional, what's the function of a muscle? Contraction. So sarcomere is the smallest structure where the contraction of muscle starts. That's why it is called the unit of muscle function or contraction. That means what? When the muscle contraction occurs, all this sarcomere inside the myofibril get shorter at the same time. So when all this sarcomere inside the myofibril get shorter, the whole myofibril gets shorter. Make sense? Because all sarcomeres get shorter at the same time, that means whole myofibril gets shorter, right? When all myofibrils inside the muscle fiber gets shorter at the same time, whole muscle fiber gets shorter. Make sense? When all muscle fibers within the fascicle get shorter at the same time, the fascicle gets shorter, right? And when all fascicles within the whole muscle get shorter at the same time, that means that's the contraction of the muscle. The whole muscle gets shorter, right? So you see where the contraction starts in the sarcomere, right? That's why it is the unit of contraction or muscle function. Okay. So you see that how beautifully the skeletal muscle is designed, right? So shortening occurs first the myofibrils, sarcomere, then myofibrils, then muscle fibers, then fascicles, then the whole muscle. Okay, so that's how the muscle contraction occurs. Now, how a sarcomere gets shorter? You need to know that. The mechanism of shortening of the sarcomere. Inside the sarcomere, you have proteins. Those proteins are very important for muscle contraction. So you need to know what are those proteins. You have protein filaments. Two types of protein filaments are present inside the sarcomere. One is called thick filament and another is called thin filament. So, thick and thin filaments. So, these are the filaments, two types of filaments inside the sarcomere. Okay, so here you see Z discs. First say Z discs, and in between two Z discs you have the sarcomere, right? So if you look at the upper picture, you see two types of filaments are present inside the sarcomere. The red colored and blue colored. Okay, the blue colored in this picture are called thin filaments, and red colored are thick filaments. I did opposite here. Okay, so anyway, so this is thin filament and this is thick filament. Okay, two types of filaments are present inside the sarcomere. You see here, thin filaments are directly attached to the Z disc, right? Thin. Thick filaments are not. How the thick filaments are attached to the Z disc? By spring-like protein structure, like this. Spring. Protein springs, like this. So these protein springs are called titin. T-I-T-I-N. Titin. Some people said titin. So you see in the picture, you see the yellow structures in the bottom picture. Thick filaments are connected to the Z disc by those yellow spring-like protein structures. Those are called titin. Okay. So these filaments are very important for the contraction of sarcomere. First, we'll see the structure of a thick and a thin filament. If you see a thick filament consists of many golf club like structures bundled together. So many golf clubs, you have seen golf clubs, right? Golf clubs. So if you bundle many golf clubs together, that looks like the thick filament. So many golf club like structures are bundled together, and that is the thick filament. Each golf club structure is called a myosin. So many myosin bundled together to form the thick filament. Okay, thick filament. Now, if I see one myosin, this is the myosin. It has a head and a tail part. Okay, head part and a tail part. Now, in the head, this is very important. In the head, you have some binding sites. This is one binding site. This is called site for actin. There is another binding site that is called site for ATP. And there is another binding site that is called ATPS. ATPS is another binding site. So there are three binding sites: site for actin, site for ATP, site for ATPS. Okay, that's the myosin head. And many myosin bundled together to form the thick filament. Now we'll see the thin filament. If you see the structure of a thin filament, it is formed by two chains. Two chains are together, that is thin filament. And the chains are formed by many round protein structures. Each round protein structure is called an actin. So many actin balls join together to form two chains, and those two chains together form the thin filament. Okay, the double chained structure, and formed by the chains are formed by actin balls or molecules. Okay, so this is the structure of a thin filament. Now, if I see one actin ball, on the top of the actin molecule, you have a binding site. This is called site for myosin. Site for myosin. Make sense? So now you see here, on myosin head you have site for actin, on actin you have site for myosin. That means what? This area will bind with this area, right? This is site for actin, this is site for myosin. So they have site for each other, right? Yes.
So that bigger circle is just one of the smaller ones in the chain?
Yes, yes. So actually it is like this, you see. The top of each actin you have site, binding site for myosin, make sense? Like this. Binding site for myosin. And each head you have binding site for actin. Binding site for actin. So that means what? These are myosin heads here, and these are actin balls. Make sense? And you have binding site here, binding site here, binding site here, binding site here. Make sense? So they will get attached to each other because site for each other. Make sense? And that happens during muscle contraction. The myosin heads will bind or get attached on the actin balls. Is it clear? This side? Got it? Okay. That is needed. That binding is needed for muscle contraction. Okay, so now what happens, you see, the binding is not that easy because in normal resting condition, when you are in relaxed, your muscles are in relaxed condition, a protein structure, another protein structure, it looks like a rope. That rope-like protein structure, that is called tropomyosin. This tropomyosin is a rope-like protein structure that covers the actin balls. Covers the actin balls like this. This is the rope, tropomyosin, this is actin ball. Masks the binding sites on the actin balls in resting condition. So the myosin heads cannot bind with the actin balls. Make sense? Because separated by that rope-like structure, which is called tropomyosin. And another protein structure that is called troponins sit on that rope. These are called troponins. So on the tropomyosin you have many troponins. So all these are protein structures. All these are protein structures. Actin, myosin, troponin, tropomyosin are proteins. Now, what happens? Now you've got the structure here. Now what happens during muscle contraction? The calcium is released, and that calcium molecules get attached to the troponins like this. So these green are calcium. Calcium is released when we decide we will contract the muscle. Calcium is released, and the calcium molecules get attached to the troponins, bind with the troponins. And when the calcium bind with the troponins, what happens? All these troponins move to the side like this. Move to the side. So when all these troponins move to the side, then the rope, which is the tropomyosin, also moves to the side, because troponins are sitting on tropomyosin, right? So you see here, this is tropomyosin, troponins are sitting on it, and if all those troponins move to the side, that will also move the tropomyosin. Make sense? Because they are sitting attached on it. So when tropomyosin will move to the side, the binding site on actin balls will get free, unmasked. So the myosin heads are waiting for that. So the myosin heads will quickly bind on the actin balls. Make sense? Because now the topomyosin has moved away, so binding site got free, right? So myosin head is here, quickly bind. Make sense? And that binding is called cross bridging. That binding is called cross bridge. Cross bridging. Okay. Remember that. Okay. So now the myosin heads are attached on the actin balls because the rope has moved away. So the heads, all these heads are now attached on the troponins. That is called what? Cross bridge. After that, what will happen is very interesting. You have a binding site for ATP, right, on myosin head. So ATP will come and will get attached to the myosin head because there is a site for ATP. ATP, site for ATP, right, on myosin head. So now that ATP will come and get attached. Yeah. So ATP gives what? Energy, right? ATP gives energy. You know that. So ATP will come. You see, look at me. This is actin ball. This is myosin head, right? Got attached. That is called what? Cross bridging. After that, ATP will come and get attached to the myosin head, right? To give what? Energy. To do what? To bend the head, flex the head, flexion of the head. Make sense? That will occur. To move the head, you need ATP. And now, when all these myosin heads at the same time will bend like this, that will move the thin filament. This is thin filament. This way. Okay? Because all heads are moving like this at the same time. So the filament will move like this. Okay? Same thing will happen from this side. So this is, you see, the myosin, this is actin. So we'll bind like this and we'll move this way. This side, same thing will happen. The myosin heads will bind with actin balls and move that way. So the thin filaments, all these thin filaments will move towards each other. That will move the discs, Z discs, towards each other, towards the center of the sarcomere. Because from this side, we move this way, from this side, the thin filaments will move this way. That will move the Z discs towards each other. And that is the shortening of the sarcomere. Make sense? So that's how all these sarcomeres will get shorter at the same time. That will make the myofibril shorter. All the myofibrils will get shorter at the same time within a muscle fiber. So the whole muscle fiber will get shorter. All the muscle fibers will get shorter at the same time. That will make the fascicle shorter, right? And when all the fascicles will get shorter at the same time, whole muscle contraction. That is the shortening of the muscle. That will be, okay? So that's how the muscle contraction occurs. Now, how many proteins you've got here? You got, first tell me how many filaments you have within the sarcomere. 2, right? Thick and thin. Now, thick filament consists of myosin, right? Golf club. And thin filament consists of what? Actin balls, right? And actin balls form how many chains? 2, right? That is the thin filament. And now you tell me, on myosin head you have binding sites, right? So one site is for what? Actin. Another site is for ATP. You remember I showed another site, ATPase, that is for the enzyme that will break the ATP. To release energy from ATP, you have to break it. Okay, so that's why it is ATPase. That's the enzyme that breaks ATP. Okay, to release the energy. Anyway, so those are 3 binding sites on the myosin head. On actin ball, you have only 1 binding site, right? That is the site for what? Myosin. So myosin head will get attached to the actin ball because they have site for each other, right? So they love each other. They will get attached to each other. Now, what is the problem? Problem is there is a rope-like structure, right, in between, tropomyosin, that is blocking, right, preventing the binding. So you have to do what? Move that tropomyosin away, right? So what will do that? The calcium. On the tropomyosin, you have troponins, proteins. When calcium will be released, will bind with the troponins. So move the troponins to the side. That will roll the rope to the side. Make sense? Now the site got free. So myosin head will quickly get attached. That is called what? Cross bridging, right? And then ATP will come, get attached to the head because there is a site for ATP, and ATPase will break the ATP, will release the energy that will cause the bending of the head. Make sense? That is the power stroke. The bending of the myosin head is called power stroke. Okay. Flexion of the head or bending of the head is called power stroke. Okay. Now if you see this picture, you will understand. You see that yellow rope in the bottom right, that's the tropomyosin, and on tropomyosin you have troponins. On actin you have site for myosin. On myosin head you have site for actin, ATP and ATPase. Okay. Now inside the sarcomere you have those structures. Another thing, just know for your information, actin and myosin, these are called contractile proteins. All these are proteins. These are called, these two are called contractile proteins, and troponin and tropomyosin, these two are called regulatory proteins. Okay. So actin, myosin are contractile proteins and troponin, tropomyosin are regulatory proteins. Now, you remember I said that to move the troponins to the side, who should be released? Calcium. Remember that? Calcium should be released. So from where the calcium comes? You need to know that, right? From where the calcium comes. On the myofibril, this is the myofibril, right? On the myofibril, you have a structure. It looks like many tubes are attached to each other. Many tubes are attached to each other, and that structure is around the myofibril. This is called sarcoplasmic reticulum or SR. Sarcoplasmic reticulum around, covers the myofibril, but it is like many tubes are attached to each other. Network of tubes. So that wraps the myofibril, around the myofibril, and those tubes are filled with calcium. So sarcomere sarcoplasmic reticulum is a network of tubes attached to the myofibril, and they are filled with calcium ion. Okay, so that is the structure that stores calcium around the myofibril. That means around the sarcomeres. And when the signal arrives here, that now you release the calcium, the signal must come, right, from somewhere. Right? When you decide you will contract this muscle, you must send signal from the brain to the heart. What is happening there? Let's close it. Okay, so when you decide that you will move this muscle, you send signal from the brain, right, to the muscle, and that is the signal coming here and telling the sarcoplasmic reticulum to release calcium. So that calcium from here is taken to the sarcomere by a tube that is called T-tubule. Okay. The T-tubule is the tube-like structure that takes the calcium from the sarcomere to the sarcoplasmic reticulum to the sarcomere. So calcium will be released here, and that will bind with the troponin. We'll move the troponin to the side, and that is how contraction will occur. Okay. So the signal will arrive here that will tell SR, sarcoplasmic reticulum, to release the calcium into the T-tubule. The T-tubule will bring the calcium and release it inside the sarcomere. Okay. Sarcomere, because calcium must bind with the troponins, right? Those proteins. Okay. So now you know what is sarcoplasmic reticulum. You remember endoplasmic reticulum inside the cell? Many tube-like structures are connected to each other, right? That is called endoplasmic reticulum inside the cell. But sarcoplasmic reticulum is around the myofibril, okay? But tube-like structures connected to each other. Here you see that blue structure is the sarcoplasmic reticulum, and it is filled with calcium. Sliding filament model of contraction. Now you remember when I told you that the actins from both sides are moving towards each other like this, and the thick filament is in the middle and thin filaments are moving like this. And what happens, this is a thick filament, this is a thin filament is moving this way, from this side this way. So that is called sliding filament model of contraction, because it looks like under the microscope when contraction occurs that thin filaments are sliding over the thick filament. Okay, so it looks like, you know, all thin filaments are sliding over the thick filament. That's why it is called. Now, you see here, the upper picture is showing a sarcomere between two Z discs, you see there, in relaxed condition. So the thin filaments, the blue filaments, are away from each other. There is a big gap in between, in the center of sarcomere. Lower picture is showing a fully contractile condition. So you see that thin filaments, the blue filaments, move towards each other and got very close. Okay, and the sarcomere gets shorter. That means two Z discs are now close to each other, closer than the relaxed condition. So shortening of sarcomere. Now, in regular microscope, if you see a sarcomere in relaxed and contractile condition, how it looks like. You see the topmost picture you see some bands or areas, dark and white. Why is that? That is very simple. When we see something under the microscope, we pass what? From the bottom? Light, right? We pass the light from the bottom. See, right? You have seen that. So, when we put a sarcomere on the slide and pass the light from the bottom, in those areas you have more structures, light will be blocked more, right? Those areas have light structures, less structures, light will easily pass. That's why you see the dark and white areas. White areas means what? You have less structure there, so light is easily passing, right? Dark means light is being blocked. Okay. Now, if you look just the areas, let's see this area. You have which filaments here? Both thick and thin, right? They are both thick and thin. So light will be blocked more there because you have both filaments, right? Now you see in this area you have only thin, right? So light will easily pass. So you see light. Here you have only thick. So not too dark, not too light. So that's why you see the bands, dark and light bands. Make sense? More structures will block more light. Okay. You don't need to remember all these name of those bands. Just know why you see those bands. Okay. They have different names, those bands. Okay. Now I'll talk about a neuromuscular junction. Neuromuscular junction, the name is telling you it is the area where the neuron talks to a muscle fiber. The area where a neuron ends on a muscle fiber. That's the junction. So if you see, first let me show you a neuron. This is a neuron, mononeuron. This is the axon terminals. Cell body of neuron, dendrites. You already know this structure. So this is a motor neuron. We send signal to the muscle through the motor neuron, motor command to contract the muscle. So the signal goes this way, and these are axon terminals. These are axon terminals, and the axon terminals end on the muscle fiber. So this neuron has, for example, 3 axon terminals, and they end on 3 different muscle fibers. Okay, so 3 muscle fibers are getting signal from this motor neuron, and this whole structure is called the motor unit. Motor unit. That means a motor unit consists of a single motor neuron and the muscle fibers innervated by it. Make sense? The muscle fibers getting signal from it. So this motor unit has how many muscle fibers? 3, right? So 3 muscle fibers and this neuron, motor neuron, together is called what? Motor unit. In your body, you have some motor neurons, those are supplying or innervating about 1,000 of muscle fibers. Those are large motor units. You have some motor units, those are supplying only 2, 3 or 4 muscle fibers. Make sense? So those are small motor units. So one single neuron can supply signal or give signal to thousands of muscle fibers too. Those are very large motor units. Anyway, so that is the motor unit, the neuron and the muscle fibers getting signal from. Now, this area where the nerve or neuron ends on the muscle fiber is called a neuromuscular junction. Very microscopic, right? This only this part. This is called a neuromuscular junction. Makes sense? The connection between the neuron and the muscle fiber. Now, if I just only see the structure of a neuromuscular junction under very powerful electron microscope, how it looks like. Okay. So now I'll see only this part under very powerful microscope. So this is the axon terminal, and this is the muscle fiber. So muscle fiber and axon terminal. Okay. When you see under very powerful microscope, what we see is that axon terminal is not actually directly attached to the muscle fiber. There is a tiny gap. Okay, and that gap is called cleft, synaptic cleft. You know, cleft means gap, right? You know, cleft lip, cleft palate. Have you seen that? Gap. So, synaptic cleft is the gap between the axon terminal and muscle fiber. And this end is called, the axon terminal is called pre-synaptic terminal. Pre means before. This is synapse, presynaptic, before synapse, the terminal. And this membrane of muscle fiber is called postsynaptic membrane. Postsynaptic membrane. Presynaptic terminal, postsynaptic membrane, and this part of presynaptic terminal is called presynaptic membrane. So very easy. Presynaptic membrane. So presynaptic membrane, postsynaptic membrane. Presynaptic terminal. Now, inside the presynaptic terminal, you have vesicles. You know, vesicles are round membranous structures, and these vesicles are filled with a neurotransmitter or neurochemical that is called acetylcholine, ACH. ACH or acetylcholine. Have you heard it name? Acetylcholine or ACH in shortcut. Okay, it is a neurotransmitter or neurochemical. That is the presynaptic terminal. On postsynaptic membrane, here you have many receptors. These are ACH receptors. That means receptors for acetylcholine. or acetylcholine receptors on postsynaptic membrane. Okay, now that is the structure of a neuromuscular junction. Now what happens when a signal, we know we send signal through the nerve, so signal comes this way, that I showed you, signal comes this way and goes to the muscle. So signal comes, electrical signal, of course, right? Nerve transmits electrical signal. So electrical signal will go this way, like this, quickly, very fast. And when the electrical signal passes through the presynaptic membrane, the channels get opened in presynaptic membrane. These channels are calcium channels. So what will happen? The calcium ions from here in presynaptic cleft will get in. So calcium ions will get in quickly, because those are calcium channels or calcium gates. Only calcium can pass through it. Make sense? On membrane, cell membrane, you have many different types of channels, right? Gates. Some are for calcium, some are for potassium, some are for sodium, right? So these are calcium channels that will get opened. So calcium will get in. Why calcium should get in here? This calcium is not that calcium we talked about before, like troponin binds with calcium. That's a different calcium, okay? So this calcium is different. So the calcium will get in to rupture the vesicles. Calcium is needed to rupture the vesicles. When the vesicles will be ruptured, ACH molecules will come out. Make sense? Enter into the cleft. So now ACH molecules have been released from the vesicles. Make sense? And these are what? ACH receptors on postsynaptic membrane, right? So they will bind with ACH because they are ACH receptors. Your name is telling you, right? ACH receptors. So these ACH molecules will bind. If other molecules come, another type will not bind. So this is like key lock system. So ACH receptor binding will occur. That binding is very important. It's like you are putting the key in the lock. Make sense? Key is ACH and lock is the receptor. Make sense? So when that fit will be perfect because these receptors are for ACH, right? So that will do what? Open the doors here. So that binding, ACH receptor binding, will open the door on postsynaptic membrane. That is very important. That those doors must be open. So when these doors will open, sodium will get in because these are sodium doors. These are calcium doors or channels. These are sodium doors. So sodium will get in from that cleft. Now sodium is here. Lot of sodium will influx into the muscle fiber. Okay, so just if you understand this part, that is very important. So let me quickly review. Okay, so this is a neuromuscular junction, and this is the axon terminal, also called presynaptic terminal. Make sense? Inside the presynaptic terminal, you have these round structures. Those are called what? Vesicles, right? Round membranous structures are called what? Vesicles. And these vesicles are filled with what? ACH. And on postsynaptic membrane, you have the receptors, ACH receptors. Is it clear? That's right. Got it? ACH receptors. Remember, I will ask you. Okay. On postsynaptic membrane, you have ACH receptors, right? Now, when electrical signal, what kind of signal? Electrical signal, right? You know, nerve transmits electrical signal. Will pass through presynaptic membrane. Which channels will get opened? Everybody. calcium channels will get open, right? So calcium will get in to do what? Rupture the vesicles, right? That will release the ACH into the clot. Is it clear? And these receptors are what? ACH receptors. So ACH molecules will bind with the receptors, right? That's like putting the key in the lock, right? That will open what? Sodium channels. Is it clear? So when sodium channels will get opened, sodium will get in. Is it clear? Sodium ions will get in. Okay. Now you see, this is interesting. If this is the muscle fiber, sodium got in. Plenty of sodium ions got in. So inside the muscle fiber, when the sodium is getting in, sodium is positive or negative? Positive, okay. So when sodium is getting in, that means positive is getting in, right? So inside the muscle fiber, it will be more positive. Is it clear? The potential will be more positive. Now if I draw a line, before sodium got in, the channels are closed. So this is the line I am showing the membrane potential, okay? And this way, if I want to show positive, this way negative, when sodium gets in, I am looking inside the muscle fiber. When sodium is getting in, inside the muscle fiber, becoming more positive. So this will move like this. That is called depolarization. So influx of sodium moves the membrane potential towards positive. That is called depolarization. I don't know if you have heard this term, depolarization. That's the influx of sodium that moves the membrane potential towards positive. And then after that, what will happen? The sodium channels will get closed and potassium channels will get open. Now remember, potassium will get out. When potassium channels will get open, potassium will get out. Potassium is also positive. So positive is getting out now. So inside the cell, remember we are always looking inside the muscle fiber or cell. So if potassium is also positive and potassium leaves, now inside will be what? Move towards negative, right? Because positive is leaving from inside. So it will move towards negative. And that is called what? Depolarization. So depolarization, everybody, depolarization is influx of what? No, influx of sodium, right? That is making inside what? More positive. Is it clear? And repolarization is efflux or outflux of potassium. Both are positive, sodium and potassium. Now think, if sodium gets in, inside will be more positive. If potassium leaves, inside will move towards less positive. We can say less positive or towards negative. So, makes sense? Very simple. Like you are the body, if I push sodium in, which is positive, your blood will move towards positive more, right? If I take positive out, it will move towards negative or less positive. So, very simple. Depolarization and repolarization. Now, if I ask you, depolarization occurs due to influx of what? Sodium. And repolarization occurs due to efflux of potassium. Is it clear? Okay. So, some channels get closed, some channels get opened at different times, and that's how those things happen. Not all the channels are open at the same time or closed at the same time, right? So that is very important. Now, the last thing is why you need to know depolarization and repolarization. Depolarization is the signal. Now let me show you. You will understand. This is the myofibril sarcomeres, tube-like structures around the sarcomere or myofibril. That is called what? Sarcoplasmic reticulum. Remember that? Filled with what? Calcium. Right? That calcium must enter into the sarcomere by through T tube. Now, I told you that a signal comes here to tell the sarcoplasmic reticulum to release calcium, right? Only that signal should go to the muscle when you decide to contract the muscle. Is it clear? That will release the calcium. So that signal is what? Depolarization. So when depolarization will occur, sodium will get in here. This is the muscle fiber, right? Muscle fiber. When sodium will get in, depolarization will occur inside the muscle fiber. That will give the signal to SR, because these are inside the muscle fiber, right? Myofibrils, to release the calcium. So very important. Depolarization must occur in the muscle fiber to give signal to SR to release the calcium into sarcomeres. So here, this is the muscle fiber. You have the myofibrils, many rod-like structures. All these are covered by SR, filled with calcium. So that depolarization will give signal to SR to release the calcium. Okay, so that's the complete picture now we got, right? So that's how the muscles contract. Okay, so now if I go back, actually I started from the smallest, right, to largest. So now if we go from largest to smallest, first you decide that you will contract a muscle, right? Your brain sends what kind of signal? Electrical signal, right? Through the nerves, right? Through the neurons. And that electrical signal will pass through the presynaptic terminal and will pass through presynaptic membrane. That will open the calcium channels. This calcium ions will rupture the vesicles. Is it clear? To release what? ACH into the cleft, right? Into the gap. And that ACH will bind with the ACH receptors on postsynaptic membrane. Clear? When that binding will occur, that will open the sodium channels, right? Doors. So sodium will get in, right? And inside the muscle fiber, you have many myofibrils, right? Dots. Myofibrils. Now remember the structure: whole muscle, muscle fascicles, muscle fibers, myofibrils. Okay. So inside each muscle fiber, you have many myofibrils. So as sodium will get in, that will cause depolarization. That will tell the sarcoplasmic reticulum wrapping the myofibrils to release the calcium into the sarcomere. That calcium, this is different calcium than this calcium, right? This calcium is here on SR. We'll get in, we'll bind with troponins, we'll move the troponins to the side, that will move the rope to the side, tropomyosin to the side, free the actin, right? Free the binding sites on actin, right? And myosin head is waiting for that, right? So that will occur. That is called what? Attachment. Cross bridging, right? Very good. Cross bridging. And then ATP will come, right? To give what? To do what?
Bend.
Bend. That bending is called what? Or stroke. Stroke. Have you seen rowing the boat? Many people do what? At the same time hit the paddle on water, right? So exactly same. Many myosin heads are hitting the actin balls like this at the same time and getting released and doing again, getting released, doing again. Make sense? So like rowing, hitting, taking out, hitting again. Doing like this, releasing again. Now, you see, to bend the myosin head, you need ATP, right? But also to release it, you need what? ATP. Because both are movement, right? To move this way, you need ATP. I did not talk about that. After that, it should be released. You need also ATP.
What is S?
ATPase is the enzyme that will break the ATP to release the energy. So both ways you need energy. Is it clear? So to hit you need energy, to take it out you need ATP. So in both ways you need ATP to move the head. Is it clear? Okay. Now after death the production of ATP stops, right? Because in non-living body, ATP will not be produced. To produce ATP, you need metabolism, right? So, after death, no ATP are produced in the body. So, what happens? Many myosin heads stay locked, cannot get released, because to release, you remember, ATP is needed, right? So cannot get released because of lack of ATP, and that causes stiffness of the muscle. Muscles remain stiff. That is called what? Rigor mortis. Right? Have you heard that? Rigor mortis or rigor mortis. So stiffness of the muscles after death. It stays like 4 to 24 hours after death. That is rigor mortis. Due to lack of ATP, the myosin heads cannot get released, come out. They stay like this, locked. Clear? Okay. There is a clinical condition that is called myasthenia gravis. Have you heard that?
No.
Myasthenia gravis occurs due to the distraction of postsynaptic ACH receptors. Myasthenia gravis. I'm sure some of you have heard this. Myasthenia gravis is a clinical condition in which the patient suffers from extreme fatigue, weakness of muscle. That happens due to the distraction of ACH receptors on postsynaptic membrane. You now understand why. If postsynaptic membranes don't have receptors, then even ACH molecules are released here, they will not find the receptors, right, to bind with, because receptors have been destroyed. So there is no lock there. So the doors will not get open. Is it clear? So sodium will not get in, depolarization will not occur, muscle will not get any signal to contract, because depolarization is giving the signal, right? So muscle contraction will not occur due to distraction of ACH receptors on postsynaptic membrane. And that distraction usually occurs due to autoimmune disease. Your own body produces antibody against these receptors. Make sense? Autoimmune. So your body is producing antibodies that destroy the postsynaptic receptors. So that's a serious clinical condition. Okay? The patient cannot move. There are a couple other conditions related to muscle. One is called dystonia. Irregular contraction of muscles. Dystonia. Irregular contraction of or abnormal contraction of muscles. Another condition that is called muscle dystrophy. You know that? Yeah, muscle dystrophy is actually many muscle fibers get destroyed or die. Okay, so the muscle dystrophy could be many different types. But just know that in muscle dystrophy, it is a genetical disorder. And mutation of certain gene that is responsible for the synthesis of protein for the muscle fibers, right? So that mutation of that particular gene that is responsible for the protein synthesis for the muscle are not present. Genetically inherited genetic disorder. So in person's body, the muscle fibers will not be produced. So another is muscle cramping. You know that you have heard, you see, right? Cramping of muscle that can cause due to different ways. One is dehydration can cause muscle cramping, right? Electrolyte imbalance, if potassium or sodium, those ions or electrolytes concentration goes down, depletion of sodium or potassium, that can cause muscle cramping. Actin myosin, remember the binding, if ATP production decreases, that will also cause that. Okay, so those are the reasons why the cramping can occur. Okay, abnormal state of mind can also do that. Anyway, so just those are a few things. Now, in your exam on Tuesday, I may ask you to draw a neuromuscular junction. Okay, draw a neuromuscular junction and show different structures inside it. Okay, so you will draw exactly what I showed you here beautifully. Okay, probably, I am not sure if I'll ask you to do that. Another thing is I may ask you to write few clinical conditions related to the muscle, right? So you'll learn those few things. Dystonia, muscle dystrophy, myasthenia gravis, right? Those things, and I'll ask you to write them. Yes.